Honeybees are overachievers. They accomplish a great deal with a very tiny brain. They're well known as exemplars in the field of communication. They are the only species outside of our own with a symbolic language. They're also known for their pollination, their contribution to our economy, contributing some $15 billion a year in agricultural productivity in the United States alone. They're also paragons of social organization, and that's what I want to focus on today, talk a little bit about one aspect of their social organization and explain to you how we've understood this, come to understand this as a function of a great deal more plasticity than one might have thought, and then delve more deeply into the molecular basis for this. So the honeybee model has a couple of attributes. First of all, normal behavior can be easily observed and manipulated. They're big enough to be tagged individually with these tags. We're now working with barcodes and so on and so forth to be able to automate these kinds of activities, but they can be tracked as individuals. And then perhaps more importantly, from the perspective of the issues of this conference, it's possible to engage in social engineering on a grand scale with honeybees. So each of these stacks of white boxes that you see here is a bee colony. It's a family. It consists of anywhere from 20 to 50,000 individuals. They're all related. There's a queen. And it's possible to then conduct social engineering experiments where we can change the social environment of the individuals in the colony with great precision, replicate that to a large level, and as you can see with the number of stacks here, though each of them is being colonies, replicate this under natural conditions, and then study the effects at different levels of biological organization. So it's possible to understand how colonies interact with their environment, how the behavior of individual bees gives rise to a smoothly functioning colony, and then down the levels of analysis, brain and gene, and so forth. So what I want to talk about today has to do with one particular aspect of the social organization of a bee colony, and that is the division of labor. So what I want to tell you about starts with the sentence that you see there, where we're going to be talking about worker honeybees. Now, worker honeybees do not grow up and reproduce. That's the job of the queen to reproduce. But they do grow up. They go through a process of behavioral maturation, changing the occupations at different ages. An adult worker honeybee lives about 6 weeks, and she spends the first 2.5 to 3 weeks working in the hive, and then she graduates and becomes a forager. Now, many of you would be forgiven for thinking that despite what seems an interesting pattern, it must be robot-like, choreographed internally in a very strict way, or as Russ Fernald found a beautiful quote from Spalding, such as music emerging from a music box just intrinsically. But I'm here to tell you today that it's a little more complex than that, and a little more flexible than that, and making it more relevant to many of the interests that you have. And that is that it's socially regulated. Bees are not little robots where they do the same thing at a particular age no matter what the conditions. It's actually, to take a quote from Clyde's talk yesterday, it's an emergent property based on social interactions. How fast a bee grows up, what occupations it takes up, actually is an emergent property based on social interactions. So what I'm going to do in the talk today is go through some older data that demonstrate that this process of behavioral maturation is socially regulated. And then in the second part of the talk about some recent molecular and analyses drawing largely on systems biology approaches to understand the molecular regulatory basis for this plasticity. So adult worker honeybees, as I said, live about 6 weeks. They spend the first 2.5 to 3 weeks working in the hive, engaged in such activities as taking care of the larvae. This is called a nurse bee. I'll probably be saying that several times. Then at about a week to 10 days, the bee makes a transition to middle age, caring for the structure of the hive and processing the food and so forth. And then, as I said, at about 2.5 to 3 weeks, she makes her final transition in life to become a forager. So you see the cartoon cartoon has a little picture of pollen and a nectar pail. This is a compass there. Prior to becoming a forager, bees take orientation flights where they learn where they live, they learn their address, they learn the direction that the sun travels. They link that by means of their circadian clock so that they can orient using sun compass bearings and then use that information to communicate by means of the dance language that I alluded to before. So this business of having a flexible pattern of behavioral maturation is what's required if you're going to make a living as a bee in a bee colony. And the reason for that, there are actually several reasons. One I'll highlight today, and that is that the colony, just like our own human societies, has shifting demographics. So there are changes in birth rates and death rates such that an individual of a particular age may find herself at one point in time among the youngest in the colony, and later on in the life of the colony among the oldest. And let me just point out to you this pink bar here, the green bar here. These are bees that are the age of, no, I want to do the white bar, that's easier, sorry. The nurse bees here, at this point in time in the spring, there are relatively few of them relative to the older bees in the colony, whereas later on here you see a very different pattern for the proportion of individuals. So if you're a 10-day-old bee, it ill behooves you to have a fixed program of behavior because you need to respond to changing colony conditions. And that's in fact what happens. Bees can speed up, slow down, or even reverse their pattern of behavioral maturation. If you remove all the foragers from a colony, some of the young bees can speed up their maturation and become precocious foragers. If you have a shortage of young bees, you'll have some Peter Pan bees that never grow up, that just continue to work in the hive. And finally, perhaps most intriguingly, you can have reversion. Bees that have made the transition, they become foragers, but if there's a calamity that befalls the nursing group in the colony, some of the foragers can revert and take up those duties again. And that leads then, of course, to the question of who's in charge. So most of the time when we think of complex social organizations in the human realm, we think of centralized control, whether it's that organizational structure for NSF or the structuring of the labor market in general, where the Wall Street Journal represents, say, a centralization of information about labor trends, and then individuals can consult that in order to know what opportunities exist. So in the bee colony, it's different. The lights are on, but nobody's home. There's no centralized control. The queen was named at a time when we humans were very fascinated with royalty. If the queen were named now, certainly by my 18-year-old very irreverent son, she would get nothing better than, yo, egg-laying mama. That's it. And truth be told, that's all she deserves, because that's what she does. She specialized on laying eggs. She does secrete some pheromones that have tonic effects on the activities of individuals, but she doesn't tell them what to do. The workers engaged in a process, a decentralized process, that tells them what to do, and I'm going to tell you about some aspects of that right now. So what we found is that there's social inhibition of behavioral maturation. Old bees inhibit the maturation of young bees. And then once you hear that, it makes perfect sense. The more old bees in the colony, then they're taking care of the foraging activities. There's no need for the nurse bees to speed up their maturation. And so what we found was that there is this process of social inhibition, and I want to take you through a couple of older experiments that led us to this conclusion. First of all, here's an experiment, a large-scale social engineering experiment. Each of those dots represents a colony with an artificially manipulated age demography, and what you can see is that there's a negative relationship there between the rate of behavioral maturation and the percentage of old bees in the colony. The more the old bees in the colony, the slower is the rate of maturation, and that's revealed by the fewer or lower percentage of young bees in the foraging cohort. Another experiment, we created the world's smallest beehive with one bee in a dish, and we reared this bee for 7 days, a bunch of these bees in individual confines like this, and then we put them in colonies and compared their behavior with bees that were reared under normal conditions. So we compared isolated bees, the green bars, with the colony-reared bees, the pink bars, and what you can see is that the green bars represent precocious foraging. Bees reared under social isolation, when placed in a colony, showed precocious foraging. So there were far greater far greater numbers of precocious foragers coming from the isolated bees than from the colony rear bees. You can barely see that pink bar right there. So it was experiments like this, coupled with our knowledge that most of the communication in an insect society takes place via chemicals, that led us to search for a pheromone, and then we found that in collaboration with Yves Leconte's lab in Marseille, France, and found a pheromone that's produced by older bees that inhibits the age at onset of foraging among the younger bees. The compound is called ethyl oleate. When we put it dissolved in sugar and let the bees feed on it, we get a significant delay in the onset of foraging, measured here as the differences in the age at onset of foraging between the control and the treated bees. This work also led us to look more broadly for other pheromones, and we found that the queen and the brood also produce pheromones that are helping to calibrate the division of labor so that you have just the right number of individuals performing the jobs depending on the needs of the colony. So I've just described to you that the process is socially regulated. In transitioning to look at the molecular basis of this, what I wanted to say here is that growing up is hard to do in bees. There are a lot of changes that occur when a little bee, despite her tiny brain of less than 1 million neurons, goes through this maturation process. There are structural changes in the brain, for example. So here we have some cartoons based on Golgi stains performed by former graduate student Sarah Ferris. This was a collaboration with Susan Farbach's lab, showing an increased elaboration of the dendritic structure of the neurons in the forager brain relative to the nurse brain for one particular area, which is outlined for you here, which is the mushroom bodies, a region of the insect brain associated with multimodal sensory integration. And that's a lot of what foragers do, integrating a variety of kinds of stimuli to be able to structure their successful foraging. So when you see something like this, it should then be no surprise that when one does a microarray experiment comparing forager and nurse brain, one would find thousands of genes showing differences in expression, and that's in fact what we found. We've done this with various microarray platforms and now RNA-seq, and we find between 20 and 40% of the genes in the brain showing differences between nurses and foragers. From the gene expression data, we get again a very strong signal of the social regulation aspects of this. So let me take you through this slide. This is a slide that shows a clustering of several of the largest groups of genes showing their expression pattern. And what this pattern shows is that the expression of these genes tracks the behavioral state of the bee rather than its age. So what we have, the abbreviations here, young and old, nurse and forager. These are the bees from the typical colonies that I have been describing to you, sort of normal bee colonies. And you can see big differences in the nurses, the young nurses, and old foragers. Now, if you do the experimental perturbations that I alluded to before, such as removing foragers or removing the young bees to uncouple age from behavior, then what you see is that when given a choice between age or behavior, these genes, which represent the largest category from this experiment, track behavior rather than age. So here we have young nurses and young foragers. This is from an experiment where you remove all the foragers from a colony, and you can see that age is immaterial. It's the behavioral state that matters, and so forth here, also for the Peter Pan bees, the old foragers and the old nurses. And so I could show you a formal quantitative analysis that shows that the dominant effect was behavior rather than age, but I won't. Another example of social regulation of gene expression gets back to the pheromone. So pheromone modulates brain gene expression in a way that's consistent with its role in behavioral maturation. For this example, I took another pheromone, this one produced by the queen mandibular pheromone, abbreviated QMP, and QMP delays the onset of foraging, and it upregulates the genes that are upregulated in nurses, and downregulates the genes that are upregulated in foragers. So we have then evidence for social regulation of behavioral maturation, and we see this echoed at the level of changes in gene expression. For the rest of the talk, I want to talk about the regulation of these molecular changes. And to do this, first, I want to use this slide both as a primer on cis regulation, on the regulation of gene expression, and also to introduce one major player in what I'll be talking about. So what we're talking now about is the regulation of genes by transcription factors. Transcription factors, together with cofactors, such as are assembled here, will interact with each other and with some cofactors, and they will bind to regions in the promoter region of the gene. These are very specific regions that recognize each specific transcription factors. These are called cis-regulatory motifs, and then they will affect the expression of a particular gene. So when one sees such massive and orchestrated changes in gene expression, the first question that comes to mind is, how is this gene expression actually orchestrated at the molecular level? So given the dominance of this form of gene regulation, we turn to this first to see what kind of insights we could derive by looking at the transcriptional regulation of genes associated with behavioral maturation. So one analysis I want to tell you about is a collaboration with Saurabh Sinha, a computer scientist who has developed some algorithms for looking broadly at cis regulation and deriving general patterns from the results. So when I've talked about behavioral maturation, I mentioned the forager pheromone that we found. I've mentioned in passing the queen pheromone and the brood pheromone. We and other labs have been engaged in a search for a variety of determinants of behavioral maturation over the years, and there are quite a few. There are 11 and counting. And they can be categorized in the following ways. Some are genetic, that is, there are different strains of bees that vary in their rate of behavioral maturation. Some bees grow up faster than others. There are environmental or social factors, such as I've talked about several of these, the pheromones, QMP, BP, ethyl oleate, the one that I've showed you the data for. There are also nutritional factors that affect rate of behavioral maturation. And then going down the line, there are hormone and signaling pathways involved. Juvenile hormone, a prominent one, vitellogenin, a signaling molecule associated with juvenile hormone, and so on and so forth. We have some hints that there are some interactions among these, that is, differences between, for example, African versus European bees that have differences in rates of behavioral maturation, have a basis in a hormone difference in the hormone system. Oops, these slides are going faster than I am, and that's pretty fast. So what this led to was a question of, with all these different factors affecting behavioral maturation, the question was, do they actually work together, or are they working separately? Are there 11 and counting different ways of regulating behavioral maturation, or is there some underlying regulatory architecture? We did the gene expression analyses for all of these factors, and the results were inconclusive. There were some genes that overlapped, some that weren't, but the level of gene expression lists of genes, is too coarse to be able to answer the question. So with Saurabh Sinha, we turned to the cis-regulatory approach to see whether we could find out a better answer. So what Saurabh did is designed a program to be able, and there are other such programs, to be able to survey the promoter regions of all the genes on these lists of differentially expressed genes, comparing one state to another. That is an experiment where we treat some bees with a pheromone, some are control, we get the genes that are differentially expressed. We compare foragers and nurses, we get the genes that are differentially expressed, so on and so forth. So those are gene lists. Those become the input now for these systems biology type analyses that I'll be talking about for the rest of the talk. So the program scans the promoter region and finds the cis-regulatory motifs that are found in these regions of the genes. And then what Saurabh did was develop a meta-analysis tool to be able to see whether there are any patterns of occurrence of these motifs that would suggest some underlying regulatory architecture. And in fact, there were. There were several motif pairs that are significantly associated with fully 10 of 11 of the determinants of behavioral maturation. And I highlight a few of them here, NF-kappa B and a couple of transcription factors associated with metabolism. significantly co-occurred in large numbers of these gene list promoter regions. And CREB, an iconic transcription factor associated with neuroplasticity, and BROAD, something associated with juvenile hormone, also were significantly co-occurring over many of these different gene sets. Giving us a picture then that there is an underlying regulatory architecture there, and, or to put it more simply, that all of those different determinants, the 11 different determinants, they are acting in common. They are converging on some transcriptional regulatory pathways to regulate the rate of behavioral maturation in response to social environment. Another way to look at this, and giving us a very similar picture, was to create a brain transcriptional regulatory network. So we've done a large number of gene expression analyses, as I've just mentioned. We've done even more than I've just mentioned. I've touched upon in the previous slides the analyses having to do with behavioral maturation. We've also looked at various behavioral states associated with foraging and behavioral states associated with aggression. Altogether, we cataloged the brain gene expression for 853 individuals encompassing 48 behavioral states. This was a large set of studies funded by an NSF Frontiers in Biological Research grant with Bruce Schatz, the principal investigator. Half of it was biology and half of it was technology, developing new text mining algorithms to derive meaning from gene expression lists. I won't be talking about that part of it. So what we did in collaboration with Nathan Price's lab was to create a brain transcriptional regulatory network from all of these data. The way you make this network is you look at patterns of expression across all the 48 states for all the genes, and then just simply create a large correlation matrix for gene expression. So genes that have more similar patterns of expression across the 48 states are lumped together temporarily in a cluster of genes. Then what you do is take all the transcription factors that you have in your genome, or that you have access to in your genome. In our case, we have about a third of the transcription factors that we expect to be there based on the numbers that are present in Drosophila. The third is a conservative number because we derive all of our annotation information in the honeybee from Drosophila, and Drosophila is 300 million years away, so to be conservative, we just take the most strongest hits, the most similar sequences. So we're working with about a third of the transcription factors. But that's still quite a lot. That's about 200. So then we take, remember, we've got these genes that show these correlated patterns of expression. Then we take the transcription factors and ask the program to find the transcription factor whose pattern of expression itself can best predict the pattern of expression of one of the temporary clusters of genes. And we have a very high threshold for success for that. A predictive value above 0.8 allows us to then make that match. The match meaning a transcription factor with a set of genes. Now we've got a module in the jargon of network analysis. And so when we did that, we found that 25% of the genes were able to be part of the network. That is, for 75% of them, we weren't able to model their expression based on the expression pattern of a single transcription factor. And that was, to tell you the truth, a very pleasing result. If we had gotten like 100%, it would have been something wrong with this, because we know that the regulation of genes is way more complicated than a single transcription factor regulating the expression of the genes. I already showed you that we have combinatorial effects going on. So 25% can be modeled based on the predictions from one transcription factor. That was a pleasing result. It said that we had some things to work with, that there is some underlying logic to this approach. So what we then found when we put the network together, and this is sort of one of the ways in which these networks are depicted. This is called a cat hairball model, and so each of those dots are the genes. The most important parts are in the squares representing these triangles. The triangles are the transcription factors. We found a sort of mosaic pattern of regulation. We found 4 transcription factors that are what we call globally active. And by that I mean they were able to predict brain gene expression across the 3 different categories of behavior: foraging, behavioral maturation, and aggression. We also found some behavior-specific transcription factors. Those are in these squares, like right here, and right here. These were transcription factors that, for example, here, that could predict gene expression for foraging, but not for maturation or aggression, or these were able to predict expression for maturation, but not foraging or aggression, and so on and so forth. So who were some of these transcription factors? Well, a couple of names hopefully will be familiar to you. Broad and NF-kappa B were global regulators, one of these four predicted to regulate expression across all 3 states. And then other top regulators, Krebs, involved in maturation, which is what we saw in a totally independent cis-regulatory-based analysis in the prior slide. FTZF1 also involved in hormone signaling, and DF1 involved in vertebrates with aggression, and we found this to be regulating expression in aggression and foraging contexts. So this gives us sort of a very broad picture of expression, suggesting there's an underlying regulatory architecture, suggesting a few individual players that are worth following up. But as powerful as these systems biology approaches are, they also are so big that they are kind of diffuse, and so we need to couple them with more specific analyses. So taking the lead from these findings from Broad and FTZF1 implicating juvenile hormone signaling in the regulation of gene expression, especially with respect to maturation. We've followed that up with an analysis that I'm about to tell you about, but first I want to tell you about this. And this is just for me to remember to say that another way that we're following up is developing methods to be able to manipulate gene expression in cultured brain neurons so that we can test just at the low level the predictions of the regulatory network. That is, what genes are regulated by a particular transcription factor. All of the information I've just given you at the systems biology level is in silico predictions. We need to marry that with empirical analyses. So I'm very proud of these slides. These are our first analyses, first results, saying that we can transfect B neurons and then show expression. And the pink is what you see, the transfections here. This is a baculovirus transfection with an actin construct, so we expect to see long, spiny neurons here, or the projections of neurons, and we can. I don't know if you can see it so well in this light, but we're encouraged about this. So what we then did was focus particularly on one transcription factor related to hormone signaling, USP, and what we find is some causal effects of this gene, this transcription factor. When we knock it down with RNAi, we can delay the onset of foraging, and that's exactly what we would predict based on its pattern of expression. FTZF1 and USP are co-regulating genes. Many genes have binding sites for both of their transcription factors for each other in their promoter regions. Here's a list of genes that, a partial list of genes that's predicted to be regulated by USP. This is now, rather than the broad diffuse systems biology analysis, this is now a specific laboratory analysis where we call CHIP, where we use an antibody to USP to find then the DNA that's bound to it, and then infer using microarray analysis what genes are represented there. And so the list is very interesting. It has the foraging genes, suggesting that hormone signaling is affecting the foraging gene. We've collaborated with Marla on the foraging gene and have showed previously that it's involved in regulating behavioral maturation. For Francine's benefit, because I know she's standing there, I wanted to show that it looks like there's some histone modification and DNA methylation effects of USP as well. So what we've shown is, I think, a nice example of the dynamic genome that social information can affect brain and behavior to affect behavioral maturation in honeybees. In closing, I want to say that we find the social regulation of maturation. We find transcription factors and other genes that are implicated in other systems. How are we to think about this? This goes back to the conversation that we started yesterday morning session. And what I wanted to say was that insects are separated from humans by 600 million years of evolution. The last common ancestor is thought to be this marine flatworm, no brain, no social life to speak of. So when we're talking about these complex patterns of social organization, we then, by definition, have have to be thinking about convergence as we spoke about yesterday. This is not an homologous trait, given that the last ancestor didn't have it. However, seeing common molecular elements there suggests then that an approach that's been developed very nicely in developmental biology can really be useful in behavior, and that is the notion of genetic toolkits for behavior, the idea that there are common building blocks that are then used and reused in separate evolutionary instantiations of a particular development of a phenotype. So in conclusion, social regulation of brain gene expression underlies behavioral maturation. We see evidence for simple, direct interactions between transcription factors that are forming a surprisingly prominent feature of the regulatory architecture, and maybe the beginnings of a syscode for social behavior. Thank you very much.